Hello everyone, how are you all? It's Igcse.science.notesz. This is the second video of the Unit 1 Biology explanation video from the book. Yesterday we finished the Chemistry of Life video, this one, and today we're going to do the carbohydrates, monosaccharides, and disaccharides. Quick revision about what we know and what we actually explained a bit also. No, we did not explain anything. However, what we actually know from the before is that the organic compounds is the key to the structure and function of living things. We know this from basic O level and chemistry and biology, right? We've discussed before in organic compounds that they contain all contain carbon, right? But many they also contain hydrogen and oxygen, right? And less frequently nitrogen, such as in protein, sulfur in some protein, phosphorus, and also in some protein. We also know some types of organic compounds from basic O levels, such as the carbohydrates, lipids, and proteins, right? So we know that most of the material in our body is, which is not consisting of water, are just organic molecules, right? So as we see over here, carbohydrates, lipids, and proteins. To sum up, we talked before about the, not to sum up, like to give an example, you know glucose, right? So we said in basic O level that it's C6H12O6. As you can see over here, it's C and H and O, which means it's carbon, hydrogen, and oxygen only, as we mentioned over here, that they consist of carbon, hydrogen, and oxygen. Okay? So, basic chemistry as well is that each carbon atom can make 4 bonds, and so it can connect to 4 other atoms, and that the carbon atoms bond particularly strongly to other carbons to make long chains. Okay? And that the 4 bonds of carbon atom usually form a tetrahedral shape. This means that the carbon compounds can be rings, branch chains, or any number of 3D shapes. In some carbon compounds, small molecules, which are the monomers. So this is basically the first important keyword. Monomer is basically a small single unit. As you can see over here, Monomer is a small molecule that is a single unit of a larger molecule called a polymer. So what's a polymer? A polymer is a long chain molecule made up of many smaller repeating monomers units joined together by a chemical bond. I'll tell you something. This is a monomer. This is a monomer. This is a monomer. So this is one, right? So this is a small monomer, single unit, small one, which is monomer. Another monomer. When you put the monomers together, stick them together with a chemical bond over here, which is going to talk about now, and a chemical bond over here. It's now bigger. This is larger and large, basically. It's mixed, so this is called a polymer, and forms another one, not 1 to 3, but number. Got it? So basically, a monomer is a small molecule that is a single unit of a larger molecule called a polymer, and a polymer is a long chain molecule made up of many smaller repeating monomers units joined together by chemical bonds. That's great. Okay, so here again, let's just revise. In some carbon compounds, small molecules, which are called the monomers, bond with many similar units to make large molecules called polymers. The ability of carbon to combine and make macromolecules, which are the large molecules, is the basis of all biological molecules and provides the great variety and complexity found in living things. Third important keyword over here is the word macromolecule. Macro from its word is basically large. If you remember, in O level, we've discussed that there are two opposite words: micro molecule and macro. It can be micromolecule, micrometer, microscope. Okay, so as you can see, micro means small, while macro means large. So the micromolecule is a micromolecule is a small molecule, and macromolecules means large. So macromolecule is a very large molecule, often formed by polymerization. Oh, pardon. Here we go. Polymerization. Great. Um, let's now talk about basically types of carbohydrates. Carbohydrates is the first lesson we're going to talk about. We're going to talk also about lipids and also about proteins. But let's first talk about the carbohydrates. Basically, carbohydrates are very important to the cells. We've mentioned that the carbohydrates are important in cells as in many forms. First, usable energy source, and important in human foods around the world. They are also important for storing energy, and they form an important part of the cell wall in plants, fungi, and bacteria. Sugars, which are types of carbohydrates, and starch, which is also type of carbohydrates. I wanted to say that starch is a polysaccharide, and we're going to talk about it now and later. Pardon me. Starch over here is a long chain polymer formed of glucose monomers. So I'm just gonna give a real example currently. We said the monomer is when you have something, 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 and then you join it together with chemical bonds to form one thing. So this is a glucose, another glucose formed together to make sucrose. Okay? Starch. Starch. Okay? Okay. Let's complete now. Sugars and starch are the best known carbohydrates. Sucrose is the familiar white crystalline table sugar, and the glucose is used as a fuel by the cells of our bodies. Starch is in rice, flour, potatoes, but the group of substances called carbohydrates contain many more compounds, as you'll discover. Okay, so now let's discover which are first the basic structure of all carbohydrates is the same. They contain carbon, hydrogen, and oxygen. No nitrogen, no phosphorus, no sulfate, only carbon, hydrogen, and oxygen. There are three main groups, as we've mentioned: monosaccharides, disaccharides, polysaccharides. Before we complete, mono means one, which is single unit. Di means two, so 2 units. Poly means more than three, like three or more, like lots. So that's a long chain. Okay? Types, okay, some have more complex molecules than another. First, the monosaccharide, which is the simple sugar. Monosaccharide is only one, which is the monomer over here. For example, glucose. Monosaccharides are simple sugars in which there is one oxygen atom and two hydrogen atoms for each carbon atom in the molecule. A general formula for this can be written CH2ON. Here N can be any number, but it's usually low. Okay? I'm gonna tell you, this is the basic one, which is CH2O. But for example, let's say we're having glucose. Glucose had 6 C's, right? So you write C6 over here. For O, it's the same number, so 6. 2 times 6 is 12, so this is the glucose formula. Okay. Okay, the triose sugar, which is the N3, have 3 carbon atoms, and the general formula is C3H6O3. So over here, we wrote C3, so H times 2 times 3 is 6, O is 3, so O3, it's the same, correct. They are important in mitochondria, where the respiration process breaks down glucose into triose sugars. Okay, that's completely great. So to sum up the monosaccharide part, the first part, of course, monosaccharide is a single sugar monomer. Monosaccharide is a single sugar monomer. Okay, one single mono. Okay, and here there are, as we said, types of sugars: the triose sugar and the pentose sugar. As you can see from the name, tri means 3 here and 3. Pent means 5, so here 5. If you don't know this, you can revise them as they're very easy. Tri, quad, pent, hex. Hex is 7 here. Okay, 6. Sorry, hex is hex, sept, hept, falan. Hex is 6. Okay? Which is the hexagonal shape. Pentagon, triangle, 3, 5, 6. Okay? So, real definition over here. Triose sugar. So as we said, triose is 3, pentose is 5, and triose sugar means sugar with 3 carbon atoms. Pentose sugar means sugar with 5 carbon atoms. Okay. So again, as we said, triose sugar means sugar with 3 carbon atoms, and they are important in mitochondria, where the respiration process breaks down glucose into triose sugars. Pentose sugar, which is sugar with 5 carbon atoms, and the general formula is with the 5. And as we know, the very, very general formula is CH2O. And so when the C is 5, 5 times 2 is 10, and O is 5. Correct. Now let's complete talking about other stuff. Firstly, the ribose and the deoxyribose. Both of them are very important in the nucleic acids. The deoxyribonucleic acid, DNA, and the ribonucleic acid, which is the RNA, which make up the genetic material. Again, basically the pentose sugar are the sugars that have 5 carbon atoms, right? So, an example is the ribose, and the other example is the deoxyribose. Here, the ribose is basically a pentose sugar that is a part of the structure of the RNA, and the deoxyribose is a pentose sugar that is part of the structure of the DNA. So the pentose sugar, which is sugar with 5 carbon atoms, has 2 examples. First of all is the ribose, second of all is the deoxyribose. The ribose is a pentose sugar that is part of the structure of the RNA, and the deoxyribose is a pentose sugar as well that is part of the structure of the DNA. You can remember it this way, that the R over here is for the RNA, and the D over here is for DNA. So the DNA, which is called the deoxyribonucleic acid, is basically a nucleic acid that is genetic material in many organisms, and the ribonucleic acid, which is the RNA, and nucleic acid, which is genetic material in some organisms, not all of them, and is involved in protein synthesis. Great. That's actually great. Okay. So let's complete the hexose sugar, which is very, very easy. Basically, the hexose sugar is sugar with 6 carbon atoms from its name hex, which is 6. Okay? So the hexose sugar, for example, of course, as we know, the C6H12O6, which is basically glucose, galactose, fructose. Hexose sugar, which is having 6 carbon atoms, and the N is 6, have 6 carbon atoms, and the general formula is C6H12O6, and that's also because of the same thing, that the really, really general formula is CH2O. So when it's 6, so C6, H times 2 is 12, and O is the same, so O6, which is like glucose, galactose, and fructose. And these are the best known monosaccharides, and they often taste sweet. Okay? So the general formulae over here show how many atoms there are in the molecule and what type they are, but they do not tell you what the molecule looks like and why it behaves as it does. To show this, you can use the displayed formulae. Although these do not show all the detailed shape in the carbon chain, they can give you a good idea of how the molecules are arranged in 3 dimensions, which is 3D, and this can help you explain why biological systems behave as they do. As you can see over here in figure C, this is the pentose sugar. It has 5 carbon atoms, 1, 2, 3, 4, and 5, and it's in the 3D shape. Or even more simply, if you see over here, the alpha one and the beta, the difference between the alpha and the beta, or the alpha and the beta, is that as follows. Very, very easy difference. One difference only, which is the position of the OH. Here in the alpha, it's down, the OH is down. Here in the beta, the OH is up. Here in the alpha, the OH is down, just as here, and the beta, the OH is up, just as shown over here. In these diagrams, the position of carbon atoms are represented by their numbers only. Note carefully the different arrangement of atoms around the carbon 1 atom in A glucose and B glucose, beta and alpha, of course. So the glucose has two isomers, which are different forms. However, they're just like same structure, same atoms, same substances. However, different forms. So it's called isomers. As you can see over here, the isomer is basically molecules that have the same chemical formula but different molecular structures. So here we go. Glucose has two isomers, which are different forms, the alpha glucose and the beta glucose. The two isomers have different arrangements of the atoms of the side chains of the molecule, the OH. The different isomers form different bonds between neighboring glucose molecules. And this affects the polymers that are made. You'll learn more about it in section 4.8.5. So just let's complete this point, like talk about anything, like the next point now, because we're going to talk about this later. Now talking about the disaccharides. Disaccharide means two. Okay, di is two basically. Like the word di means two. So disaccharide is consisting of two monosaccharides joined together by chemical bond. For example, the sucrose, which is the table sugar we eat, it's formed by two molecules of alpha glucose joining with a molecule of fructose. So a glucose and fructose. Sorry. So it's formed by a molecule of alpha glucose plus fructose equals sucrose. Two monosaccharides join in a condensation reaction. Condensation reactions means the removal of water to form a disaccharide, and a molecule of water is released because it's removed, of course. Anyways, the link between the two monosaccharides, which is the chemical bond that makes the two monosaccharides bond together, is called the glycosidic bond, and this of course is a result of a covalent bond reaction. Here the condensation reaction is a reaction in which a molecule of water is removed from the reacting molecules as a bond is formed between them, and the glycosidic bond is the name of the bond, which is the name of the covalent bond formed between two monosaccharides in a condensation reaction, which can be broken down by a hydrolysis reaction, which is addition of water to release the monosaccharide units. Okay. We use numbers here to show which carbon atoms are involved in the bond. If carbon 1 on one monosaccharide joined to carbon 4 on another monosaccharide, we call it 1-4 glycosidic bond. If the bond is between 1 and carbon 6, it's a 1,6 glycosidic bond. We're going to talk about this later in the notes video. Just let me show you something. This is carbon 1, right? So OH here, carbon 2 OH here. So this is 1,4 glycosidic bond. 1,6 is more branched, okay? So when different monosaccharides join together, different disaccharides are made, and these have different properties. Many disaccharides taste sweet. So here are common ones. Sucrose, as we mentioned before, it's just like glucose and fructose. This is basically stored in plants such as sugar cane, and this is used in the table sugar, so you can remember that it's soluble and sweet. Lactose is the milk sugar. It's made of glucose and galactose, and this is main carbohydrate found in milk. Maltose is basically the malt sugar, and it's glucose plus glucose, and it's found in germinating seeds such as barley. Okay, here's the test for sugar, banana solution, etc. And we're currently done. Let's just revise the keywords. We've talked about a couple of keywords, not couple, many keywords, which are very, very fun to know because they're just like easy, basic keywords, basically. Keyword number 1, monomer. Monomer is a small molecule of a single unit of larger molecule called a polymer. This is such as glucose. Polymer is a long chain such as starch, and it's made up of many smaller molecules, repeating monomers, units joined together by chemical bonds. Macromolecules, a very large molecule. Micro, very small. Starch, long chain polymer. Sucrose, sweet tasting disaccharide formed by the joining of glucose and fructose by a 1,4 glycosidic bond. Glucose is a hexose sugar, C6H12O6, so C6 six carbons, so a hexose monosaccharide, single sugar monomer. Disaccharide means two, so it's made of two monosaccharides units joined together by glycosidic bond formed in a condensation reaction. Condensation reactions means the removal of water. Okay. Uh, okay. Polysaccharide is a polymer consisting of long chains of monosaccharide units joined by glycosidic bonds. Triose sugar with 3 carbon atoms. Pentose with 5 carbon atoms. Ribose, pentose sugar that is part of the structure of the RNA. Deoxyribose, a pentose sugar that is part of the structure of the DNA. Deoxyribonucleic nucleic acid is the DNA, which is nucleic acid that is the genetic material in many organisms. The ribonucleic acid is the RNA, which is a nucleic acid, which is the genetic material in some organisms and is involved in protein synthesis. Hexose sugar is the sugar with 6 carbon atoms. Isomer is, as we mentioned before, that it's the molecules that have the same chemical formula but different molecular structures only. For example, the position of the OH between alpha glucose and beta glucose. Condensation, as we mentioned earlier as well, the reaction in which a molecule of water is removed from the reacting molecule as a bond is formed between them. Glycosidic bond is the type of bond, a covalent bond formed between two monosaccharides in a condensation reaction, which can be broken down by a hydrolysis reaction. Hydrolysis reaction means adding of water, and this caused the release of the monosaccharide units. Reducing sugars that react with blue penetic solution and reduce the copper 2 ions, copper 1 ions, giving an orangey red precipitate. Lastly, the non-reducing sugars, which are the sugars that do not react with penetic solution. Next video will be the carbohydrates, polysaccharides, and of course we're going to make more videos for the same chapter, same unit, which is carbohydrates, monosaccharides, disaccharides, and polysaccharides, with the notes and stuff. Notes book will be uploaded and posted on the Telegram account, Igcse.science.notesz, as well as many like other notes, handwritten notes will be posted in the Instagram account. So basically Igcse.science.notesz. Okay. And that's it. Have a nice, pretty day. Bye bye.